started. On behalf of the College of Natural and Agricultural Sciences here at UC Riverside, I'm happy to welcome you to the second presentation in the 2014 Science Lecture Series. I'm Mary Drosser. I'm a professor in the Earth Sciences Department. And as I mentioned in the first lecture, the science lectures are a little bit different this year. As always, they're supported by the College of Natural and Agricultural Sciences. But support this year also comes from the Department of Earth Sciences, the Office of Research and Economic Development, and from a new institute, Environmental Dynamics and Geoecology Institute, or EDGE Institute. And the institute is unique in that it will bring together scientists from the biological, chemical, and earth and environmental sciences to examine specific questions related to life in this changing environment. And the person who will be director of the institute will hold the newly endowed chair, the William Mayhew Chair in Geoecology, which has been made possible by a $1.5 million gift by an anonymous person, but who is passionate about the ecology of the Southwest. And we're deeply indebted to these donors, especially the commemoration of Bill Mayhew. Dr. Mayhew is one of the founding faculty members of UCR and was one of three individuals who established the University of California Natural Reserves, which are such an important part of our heritage. So these are very exciting times for geoecology at UCR. And as part of that, I'm very excited to welcome Dr. Andy Knoll. Andy works at Harvard, and he has a host of titles. He is the Fisher Professor of Natural History and Professor of Earth and Planetary Sciences, and is also the curator of the Paleobotanical Gardens in Harvard's Herbaria. And honors of Andy's include, I actually have to read these because it's so long. He's a member of the National Academy of Sciences, the American Academy of Arts and Sciences, the American Philosophical Society, and the American Academy of Microbiology, as well as the Paleontological Society Medal, the Wollaston Medal of the Geological Society of London, the Moore Medal of the Society of Sedimentary Geology, and both the Walcott and Thompson Medals of the National Academy of Sciences. And he received the Phi Beta Kappa Book Award for his book, Life on a Young Planet, The First 3 Billion Years of Evolution on Earth. And I think that lots of times people think in terms of understanding the fossil record or understanding life on this planet when skeletons first appeared. But that happened only in the last 15% of Earth's history. And while Andy's done a lot of work in that last 15%, much of his work has been exploring the history of life on the early planet, which is about 3 billion years of our Earth's history, sort of as life unfolds on this planet. And often we in paleontology and geology talk about how when we go to look for life on other planets, that we've run that experiment on this planet. We've had the origin and evolution of life on this planet. And so we can look back to the early history of this planet for understanding what we should look for on other planets. So it's not a surprise, of course, that Andy has been very much involved in the Mars mission, as well as many of his own missions on this particular planet to almost all of the continents. So it's a real pleasure to welcome Andy, and it's also a pleasure, before I bring you up, to welcome the many students who are here. So we have students from the STEM Academy, students students from the Murrieta Valley High School. Norco has a couple of classes here, and Landmark as well. So welcome particularly those of you who are in K-12 and the many other students who are visiting UCR, and we welcome you to come here as your university as well. So thank you very much, and welcome Andy.
Okay, well, thank you everybody for coming out this evening. It's a pleasure to be here. And as Mary said, what I want to talk about this evening is the truly deep history of life. I think many of us, when we think about ancient life, will think about dinosaurs. And dinosaurs are very old in the sense that they've been gone since 66 million years ago and started up around 210 million years ago. But if you look a little bit more into the geologic record, you might think about older organisms that lived in the oceans, like trilobites. and those take you back maybe 520 million years or so. But things like trilobites turn out to be the end of my story, not the beginning. And what I'd like to do this evening is share with you some of what we have learned through time about the first 85% of life's history, and hopefully give you some sense, not just of what we think the facts are, but how we've actually come to make the interpretations that we do. I'd like to start off, though, with 3 historical panels to give you some sense of where we're going in terms of ideas, and recognize that the basis for the things that we talk about today actually go back, in some case, centuries. So the first person that I want to talk about just a little bit is a man named James Hutton, who was a son of the Edinburgh Enlightenment, living in the late 18th century. And Hutton is generally considered to be the father of geology. And the basis for this is the book that you see on the right-hand side of the image. It's something called The Theory of the Earth, which was first presented to the Royal Society of Edinburgh in 1785, and then published in 1788. Now, what's interesting is if you actually read Hutton's theory of the Earth, his motivation for coming up with this theory was actually explicitly biological and not geological. Maybe if I don't move, you won't hear that sound. But Hutton, like most good late 18th century naturalists, would walk around the hills north of Edinburgh, and he would notice that the trees and shrubs that he saw on those hills were very well suited for the environments that they occupied. And he went down to the Firth of Forth and found that the sea anemones and algae within the Firth were also very well suited for the waters that they lived in. But Hutton noticed something that nobody else noticed. He noticed that erosion was continually wearing down those mountains and hills where the plants lived, and that the products of that erosion, sand grains, were traveling down through rivers and being deposited in the Firth, slowly filling it in, thereby destroying the habitat of the sea anemones and algae. So the question Hutton asked himself is, how can life on this planet be maintained over long periods of time when the environments for which organisms are manifestly well suited are in a constant state of decay? And he came up with a brilliant solution to that. His solution was that there was a continuity of environments, but that it was a dynamic continuity, by which I mean that this mountain might wear down through time, but some process, and he thought it was heat, not entirely incorrect, would actually lift up other places, like the Firth of Forth, would eventually be lifted up, and its sediments would become the rocks in a mountainside. And so that while any mountain would disappear through time, there would always be the montane habitats that would support the particular plants. So Hutton gives us this sense of a planet that is in a continual state of change. Now, the next person is probably better known to most of you. About 70 years later, Charles Darwin wrote probably one of the most important books of the 19th century, on the left, The Origin of Species. And what Darwin said is that populations are no more static than environments. That populations are always in a state of change, and that in fact it was the process of natural selection that would allow populations to adapt to particular environments and perhaps track them as they changed through time. So now we have an interesting situation where both the physical Earth and the biological Earth are subject to change, and subject to change caused by the other. So environmental change might drive changes in organisms. Changes in organisms, in turn, might feed environmental change. And then finally, maybe the least expected of the triumvirate that I want to talk about to begin with is this man named Percival Lowell. Percival Lowell, at the end of the 19th century and the beginning of the 20th, had the kind of fame that Carl Sagan enjoyed during the latter part of the 20th century. He would fill auditoriums across North America and Europe to talk about life on Mars. And his ideas on this were conditioned by what he thought he saw in images of Mars. And in the upper right of that picture, you'll see the map of Mars that he drew based on what he considered to be his own observations. And you'll see there are these very straight structures crisscrossing the planet, which he interpreted as canals. And that, in fact, those were canals that would gather seasonal meltwater from glaciers at the north and south pole of Mars and supply the needs of an increasingly parched civilization at low latitudes. Now, if you actually look at the picture in the bottom, that's actually a picture of Mars taken about 6 years ago by the MER rover Spirit, and you can see that one of his predictions was correct. Mars is actually a very dry planet. Surprisingly, we actually think that Mars was a wetter planet early in its history than it is now. Not for the reasons that Percival Lowell would have suggested, but nonetheless, Lowell was one of the first people to suggest that not only do planets change by oscillating back and forth the way Hutton suggested they would, but that planets can actually have trajectories in environmental history. That Mars was a planet that was wet and it became dry. And one of the arguments I'm going to make is that Earth has a trajectory in its environmental history as well. It was once a planet without oxygen, now it's a planet with lots of oxygen. And that whole idea of long-term environmental change in planets really in some ways goes back to Percival Lowell. So if we take those three gentlemen together, what we find is that we have the makings of perhaps a very dynamic and interactive history of our planet, in which at times life will drive environmental change, at other times environmental change will drive changes in life. And so that's basically the basis of everything I'm going to say from here on in. Now, the one fact you have to know, and I think it's my favorite fact about our planet, is that our planet actually records its own history. And it does that through sedimentary rocks. And many of you have seen, whether in mountainsides in California or in the Grand Canyon or elsewhere, that much of the surface of our planet contains sedimentary rocks laid down one bed at a time through history. So, for example, the picture that you see on the screen is a cliff in Western Australia. And from that cliff, I can determine, based on the physical and chemical characteristics of that rock, that those rocks were laid down right along the edge of the ocean at a time when the Earth had very little oxygen. From some of the other features of the rock that I'll talk about in a minute, I can also tell you that there were thick communities of bacteria that were spread out across the sea surface and the surface of the sea floor at that time. And because some minerals contain radioactive isotopes that decay through time at a known rate, I can tell you that that marginal seaway existed about 1,850 million years ago, not quite 2 billion years ago. So we have this wonderful archive that our planet has left for us. It contains a record of biological history, the fossil record. It also contains a record of environmental history encrypted in the physical and chemical characteristics of rocks. So if we can look at rocks through time, we can get a sense of how Earth and life have interacted over really most of the history of our planet's surface. But it's worth pointing out that we actually have a second record from which we can infer something of our planet's biological history, and that is the record provided by the relationships of organisms alive today. So if you look at the left-hand side of that picture, this is what's called a phylogeny. That is, it's a hypothesis of the evolutionary relationships among organisms, based in this case for the most part on analysis of the sequence of bases in DNA or genes from these organisms. It's really a molecular analysis, and what that tells us is that humans are more closely related to chimpanzees than either humans or chimps are to other species. That doesn't mean that humans evolved from chimps, but rather that both humans and chimps diverged from a common ancestor. That humans and chimps in turn are more closely related to gorillas than they are to any other organism, and down through this pattern of nested relationships that's well depicted by a tree. What you see here is simply a record of primates, that is the family of mammals that includes us. But you'll see that in addition to suggesting this nested pattern of relationship, there's actually a time axis on the left. And we can actually put this in the framework of time because of things like the little guy we see on the right, which is a reconstruction of one of the oldest primate fossils known. Comes from Alabama. And if you look at that, you might say, well, that looks a little bit like lemurs or tarsiers today. And indeed it does. And that would be the prediction you would make from the comparative relationships that we see in the figure. So we have this nested set of relationships. We have a fossil record that allows us to say that the diversity of primates, our own family of mammals, has been one of diversification over the last 50 or 60 million years. Now we can broaden that out. Again, here on the left, you have a tree for really all animals, and the primates would be one branch on the right-hand limb there of mammals. And again, this suggests that mammals diverged from the group that gave rise to reptiles and birds on the order of 300 million years ago, that group in turn diverged from the ancestor of amphibians somewhat earlier, and you can go all the way back to fish, jawless fish, and even the closest invertebrate relatives of organisms that have backbones. And that takes you down about 400 or 520 million years ago. In fact, if you want to look at the earliest fossil relatives of our own large branch of the tree, the oldest fish, and therefore the oldest vertebrate animal, is that thing called Hycoichthyes in the top, which is about 520 million years old, from China. And if you look closely, you would see characters that would let you recognize it as a fish. It has a skull, it has the kind of musculature that we see in fish, it has fins, etc. The thing on the bottom is an invertebrate relative of the vertebrates, and again that's found in the same beds that are about 520 million years old. If you ask, what are the oldest things that we can look at that we think are animals, they are things found in the Ediacaran period that Mary Droser works on. So on the left we see things that I think many people would agree are of animal origin but are not easily interpreted in terms of animals we see today. On the right, on the top, are some things that we've worked on from China, which we at least would interpret as the eggs and early embryological development of early animals. And then that kind of starburst pattern that you see on the bottom. If any of you have ever had fairy shrimp or brine shrimp, where you get a little package of just dust and you put it in water, and within about 24 hours you have little shrimp-like animals swimming around. If you look at it under the microscope, it would look very much like this. Those fairy shrimp are actually encased in a coating or a spore that allows them to lie dormant in the sediments until conditions for growth are right. And we interpret similar structures in rocks that go back to about 580 million years ago as that stage in the life cycle of early animals. So the important point is that if we look at the Earth history, or the geologic time scale on the left, the end of the trail, in a sense of going backward through time for animals, is on the order of 600 million years, a little bit more from chemical evidence of animals, but not much. The Earth itself, on the other hand, is more than 4.5 billion years old. And so the question then becomes, if animals only represent the last part of our planet's history, what might we think about biological or life in the earlier history of the planet. And again, we get some predictions from comparative biology. Now what you're looking at is another tree showing relationships among living organisms. And this is what's sometimes called a universal tree because it purports to show at least a hypothesis of evolutionary relationships among all organisms, not just things that have backbones or skeletons. And if you look at that tree, again, based on molecular comparisons, there are three great branches. One is the bacteria, which are pretty much what you think they are. One is called the archaea, which is a group of very simple organisms that look superficially like bacteria, and in fact, until 1977, were thought to be bacteria, but as they started to be studied with molecular biology, it became clear that they were a very distinct group of organisms. And then the third branch, the eukarya, is organisms that have a nucleus in their cells, like us, and all other so-called eukaryotic organisms, which at least on this tree are shown as being more closely related to the archaea than either is to the bacteria. Now, I will say that there's a lot of argument about the exact branching order of organisms on this kind of universal tree of life, but the important thing for our purposes is something that people would agree on. All of that history of animals that I've sort of run through in terms of those trees and fossils is found only on that tip of that shallow branch marked by the blue arrow. Most of the history of life then is things that diverged on earlier branches, and nearly all of that is microbial. So the study of comparative biology suggests if we want to find that earlier history of life before we have a fossil record of animals, we should be prepared to look for microorganisms. And that raises a very straightforward practical question. Can we find a tractable and interpretable record of things like bacteria in the sedimentary rock record? Bacteria are tiny. They seem to us at least to be evanescent. Can they actually leave the kind of footprint in the geological record that we can interpret the same way we might interpret dinosaur bones or trilobite skeletons or something like this? Now, to answer that question, I'm going to talk about the rocks that you see in this picture. These come from the island of Spitsbergen, which lies about halfway between the northern tip of Norway and the North Pole. These particular rocks are about 79 degrees north latitude. And the beautiful thing about working up there is you don't have a lot of plants to get in your way. And so what you see there, each swatch of color that you see, the light and dark gray colors on that cliff in the background, each of that is about 1,000 feet thick. So you have this beautiful archive of rocks that it turns out were deposited between about 750 and 800 million years ago, beautifully exposed, ready for study. What are we going to find when we look for evidence of biology in these rocks? Because after all, it turns out if you actually go up that hill and go off to the left behind the nose of that Nunatak in the front, you will find evidence of early animals. But these are older rocks. They lie beneath it. They're at least 100 million years older than any rocks in which we've ever found evidence of animals. So what do we find? Well, if we look first at that top panel that you see on the right, these are limestones that were laid down in a tropical to subtropical seaway, not unlike the Bahama banks today. You'll see that there's kind of a nice swaley lamination in them, and people who study sedimentary rocks know that today, if you want to find that kind of laminated pattern, you would go to places where microbial communities make mats on the seafloor or the intertidal zone, and those mats then interact with incoming sediments. Also, if you look in the middle, there's this place where it all bows up, and that's something that geologists would call a teepee structure, and that tells a geologist that these sediments, as they were being deposited, were commonly left high and dry. that they were above the tidal zone. What happens then is you actually precipitate carbonate cements. They cause pressure to build up, and the beds just buckle. So there's two important points there. First is that the physical features of those sedimentary rocks allow a good geologist to place the formation of these rocks in terms of environment. And the second is that those textures of those swaley laminations suggest that life might have been present. Now, if you take a piece of that limestone and just cut it to paper thickness and look at it under a microscope, all you'll see are interlocking crystals of calcium carbonate minerals. But if you notice that those black pancake-like structures there, those are concretions made out of a different mineral, silica, SiO2, fine-grained quartz. Those nodules grew within the sediment soon after it was deposited. And because silica is much more difficult to recrystallize than carbonate rocks, it has resisted alteration during the time since it formed, and in fact preserves at a micron scale the textural and, as we'll see, microbiological features of the original sediment. So if you take a paper-thin section of that material and look at it under the microscope, you see something like what's shown in the bottom panel. Those are cells. Each one of them is about 4 microns long. And if the micron isn't a familiar unit of measurement to you, think that about it would take more than 100 of these cells to make lined up to make something about the width of a period in a newspaper. These are photosynthetic bacteria, a group called cyanobacteria, which does photosynthesis the same way green plants do. So that tells us something interesting. Under the right conditions, things like bacteria leave a fossil record much in the same way that dinosaurs left a fossil record in terms of their bones. It turns out that under favorable circumstances, we can actually say quite a bit about these things. So the particular characters you see on the top, these fossils again come from 750 million year old chert nodules in Spitsbergen. On the left, you'll see there's this round area that has this amorphous goo in it. That's where the cell was, but that decayed. It's about 30 microns across. But in life, that cell made a series of external envelopes made out of various polysaccharide compounds. And just as the dinosaur's bones are preserved in rocks preferentially relative to its organs, in cyanobacteria, the hard parts, the parts that are likely to preserve, are these extracellular structures. And those are preserved really quite well. These sometimes occur as individuals, but commonly they formed little clusters on bedding planes. And if you go and then look at them carefully, you can actually reconstruct a simple life cycle, which is what we see on the bottom. And we can then take that information and say, do we see things like this today? Well, remember, we know something important about these fossil populations, and we know that they lived in a warm, wet, tidal flat environment, probably in the tropics. So, fortunately, for those of us who work in places like Spitsbergen in the summer, in the winter, you can go to the Bahamas. And if you do that, what you see on the tidal flats are things like you see on the right-hand side there. Those are limestones, fine-grained limestone particles accumulating, and the black spots are actually populations of microorganisms made up of the kind of things you see in the lower left. That is, things that are morphologically identical and have the same life cycle that we inferred for those fossils. And in fact, this is a case of a newly discovered living cyanobacterium whose existence was predicted on the basis of 750 million year old fossils in a comparable environment. So we have a lot of confidence in that case that when we look in these rocks that are older than anything known to contain animal life, we see records of these photosynthetic bacteria that start helping us to build a catalog of what life was like before the world contained animals. And it turns out when you go to different environmental settings in Spitsbergen, that is sedimentary rocks that reflect deposition in different environments, you find all sorts of things. So on the right-hand side, there are various types of bacteria, cyanobacteria and other bacteria. The three on the left are all fossils of eukaryotic organisms. The one on the left is actually a small portion of what would have actually been maybe a centimeter-sized green alga. The one in the top has close relatives today called testate amoebae. Probably not something you talk about at home over supper time, but these are well-known organisms with well-known biology and ecology. And then in the bottom, that thing is just about 0.5 millimeter in diameter, large, complex cells, demonstrably from eukaryotic organisms. So that's the good news. Microorganisms, both eukaryotic and bacterial, leave a tractable fossil record that we can track backward through time. Microorganisms leave another interesting kind of record as well. If you look at that cliff on the left-hand side panel, you see all the beds nicely going upward along the cliff, and about halfway up you'll see lozenges, sort of bun-shaped structures. Those turn out to be about 5 meters thick, each one. They are made up of the kind of laminated structures that you see on the right. These are called stromatolites, and what you're looking at is reefs made by microorganisms. So just as corals make reefs today, for most of our planet's history, reefs were built by bacteria. And they make conspicuous structures that you see on the walls of cliff. In fact, these features called stromatolites are the most abundant structures in limestones deposited through the first 75 or 80% of our planet's history. They tell us that microbial populations were everywhere. How do we know how to interpret them? We know because there are places where you can find stromatolites forming today. The place you see on the left is called Shark Bay in Western Australia. If there is a mecca for people who study the early Earth, it is Shark Bay, because we can actually see and study the processes by which microorganisms trap and bind sediments and build these laminated structures. The image on the right is simply a cross-section through one of the structures you see on the left. So it's a great example of how Earth scientists study process with things that are happening today and learn how process generates pattern that we can then go back and look at patterns in the geologic record. Well, it turns out there's still other ways that life has left a record in sedimentary rocks. Most sedimentary rocks have a little bit of organic matter in them that are the remnants of biological materials that were buried in sediments and accumulated there. And just as some parts of organisms, the skeletons of a dinosaur, the shell of a clam, preserve preferentially, some molecules actually preserve preferentially in the record. These have been studied by petroleum geologists for many years. Now, the bad news is that generally speaking, DNA and proteins just don't preserve over long periods of time. I think the oldest unambiguous fossil DNA comes from some human relatives that are about 400,000 years old. On the other hand, some molecules can be preserved in sediments over billion-year timescales, and in particular, those are lipids. So, one of the things we might think about, or one of the things we find in the record, are molecules made by eukaryotic organisms called sterols. All of you make cholesterol. And I like to tell my students that when you die, the last bit of you that will be around for future generations to ponder is your cholesterol. So next time you go to McDonald's, you might consider that. So it's nice, and Gordon Love on this faculty is a real expert at extracting and interpreting that molecular record of life, which again can tell us about groups of protozoa, algae, bacteria, and archaea that might not have left a body fossil record, but have left a molecular record that we can understand. And so if you look in rocks the age of those in Spitsbergen, we find evidence of different kinds of algae, different kinds of bacteria. And then finally, individual bacteria are tiny, but they are so numerous that their activities can actually influence the composition of seawater. And so what I've cartooned on the right is a geologist's view of photosynthesis, in a way, and that is organisms take carbon dioxide and fix it into carbon in sugars that are then used to build our bodies. And what's interesting is that the carbon dioxide comes in two isotopic flavors. Isotopes, some of you will know, are different forms of the same element that have different weights. So carbon generally has a molecular weight of 12, 6 protons, 6 neutrons. All those things that make it carbon have to do with the number of protons and electrons. But there are different forms of carbon. About 1% of the carbon in this room has an extra neutron. It's called carbon-13. And because that's heavier than carbon-12, molecules that contain carbon-13 will actually move a little bit more sluggishly than carbon-12 containing molecules, and that will end up resulting in measurable differences in compounds. There's also carbon-14, about a couple of parts per trillion in this room, but that's radioactive and decays to nitrogen on timescales of thousands of years, so we're not going to see it in the deep past. So anyway, what this cartoon is suggesting is that if we have CO2 that contains carbon-12 and carbon-13 in seawater, algae or cyanobacteria will actually, in preference, use CO2 containing carbon-12 to fix into organic matter. And the result of that is that if you look at the ratio of these two isotopes, carbon-13 and 12, in organic matter that gets buried in sediments, it will actually differ from that same ratio in limestones deposited at the same place by a known amount. That's actually, it's about 25 to 30 parts per thousand, something that can be easily measured with mass spectrometers. And that turns out to be a signature for photosynthetic fixation of carbon. So when we look at these rocks from Spitsbergen, they tell us that there was a carbon cycle that was driven by photosynthesis 800 million years ago, just as there is today. And I could tell the same story for sulfur. So if you look isotopically at sulfur that's contained in these rocks, they tell you that there was a microbial sulfur cycle that had the same types of bacteria driving it as occur today. So the punchline then is that when we go back to these rocks that are older than anything that contains animals, they are full of evidence of life. There are fossils, there are stromatolites, there are ancient molecules, there are these chemical records of life. And so 800 million years ago, life may have been largely microbial, but its signature is pretty much everywhere you look. What happens when we go back through time? Let's double the age first of all. And this is a place in northern Siberia. And again, we're looking at limestones that were deposited, in this case about 1,500 million years ago. What do we see there? Well, it turns out we see much of the same things. Again, on the left, I hope some of you have noticed that right in the middle of the picture, there's a sort of a couple meter scale bun-shaped structure. And I hope some of you said, that's one of those microbial reefs. And if you did that, give yourself 5 points. It turns out, however, that if you look at my colleague Misha Semikhatov, who I must say is the perfect field companion to have, because when he has his hat on, he's exactly 2 meters tall. So he's just the guy you want for your pictures. Notice that he's actually standing on a structure that has a rounded surface. He's standing on a microbial reef that's probably 2 or 3 times the size of this room. And if you look closely, the whole cliff wall behind him is part of a microbial reef that would have been larger than this building. So as we go back through time, we see this record of microbial interactions with sediments continues to be important as we go back through time. In fact, we see reefs that are as large as reefs built by corals today, but in this case by bacteria. We also see microfossils. In the upper right is a fossil cyanobacterium, these bacteria that are capable of green plant photosynthesis. We also see in the lower left, and these are 20 micron scale bars, fossils of simple eukaryotes. Not as many as we see in younger rocks, but they're still there. If we can find biomarker molecules, that is, these lipids that preserve evidence of a wider diversity of life, and if we look at the carbon and sulfur isotopic signatures, they suggest that 1,500 million years ago we had biological carbon and sulfur cycles that can be interpreted in terms of things we see today. So still plenty of life 1,600 million years ago. Let's go right to the chase then and go back to some of the oldest rocks that we can look at and hope to find evidence of life. And I like this picture. It is actually something that happened to us when we were trying to look at these very ancient rocks in Western Australia. And what it shows you that field geologists come in three flavors. There are those people that when there's an accident or something goes wrong, get right in and try and fix it. Those are the people in the front. There are people like my friend and colleague Dave Johnson in the standing behind who watch those people fix it. And then, of course, there are people like me who photograph the other two. Now, why did we want to go to the trouble to go to this extraordinarily remote place? Well, the reason is that among volcanic rocks that are well dated at about 3.5 billion years, there are sedimentary rocks. And those sedimentary rocks, if you look carefully at the figures, you should see that they are laminated with those wavy laminations, just the way we saw in the much younger rocks. In fact, that's a 6-inch ruler there on the right. You can see just to the left of the ruler, we have these things that are bent up to make a conical structure. And I think there is a good case to be made that these, again, are stromatolites made by the interactions of microbial communities living on the seafloor 3.5 billion years old, much as we see in younger rocks. No one has ever found what I would regard as convincing microfossils in rocks of this age, and it's very hard to make a case for specific molecules, at least in work to date. But again, if you look at the isotopic forms of carbon and sulfur, they suggest, in complement to this physical record of stromatolites, that when we first start seeing a preserved record of sedimentary rocks that tells us about Earth history, there was already a biological carbon cycle and there was already a biological sulfur cycle. So that suggests that Earth has been a biological planet for most of its existence. Now, I have to say that we don't know a lot about that earliest life in terms of how it fits into the kind of tree that I showed you on the basis of comparative biology. But the one thing we know about that life, and have confidence in, is that it thrived on a world that did not contain oxygen. Now you might ask, how do you know that? We don't have samples of the atmosphere from back then, but what we do have are minerals that are actually very sensitive to the presence or absence of oxygen. So the cliff that you see in the background of this picture is something called banded iron formation. This particular example is about 2.5 billion years old from Australia. And the interesting thing about this is if you look at sedimentary rocks deposited through that first billion years of the known rock record, between about 3.5 and 2.5 billion years old, they commonly contain these iron-rich rocks. After that, that's not going to be the case. These kind of rocks cannot in principle form in the oceans today, and the reason they can't is that iron is essentially insoluble in the presence of oxygen. That is, if you have iron and you have oxygen, you will make an iron oxide and it will actually precipitate as a mineral on the seafloor. On the other hand, iron in the absence of oxygen can be transported through seawater. And what these rocks tell us is you had to have an ocean in which you could actually transport iron over long distances from its site of origin to its site of deposition, and that can only happen in water masses that don't contain oxygen. So that's our first clue. In fact, there are several lines of evidence, and again, Andre Becker in this department has done a lot of work on this issue. Several lines of evidence based on largely features of chemistry and mineralogy that are very sensitive to oxygen converge on the idea that before about 2.4 billion years ago, the atmosphere was essentially devoid of oxygen, but afterward it permanently acquired some oxygen content. I won't run through the reasons for that because of time, but I'd be glad to talk about it with anyone afterward. The important thing as far as I'm concerned is this is a revolution in Earth history, and the cyanobacteria are the heroes of the revolution. It turns out there's only one process that we know of on this planet that can generate oxygen in the quantities that will account for the oxygen that has accumulated in our atmosphere, and that is oxygen-producing photosynthesis. Oxygen-producing photosynthesis evolved once in one group of organisms. That was the cyanobacteria. And some of you might say, yeah, but algae and plants photosynthesize too. But what you should know, if you don't already, is that the chloroplasts in the oak tree outside on campus are themselves derived from once free-living cyanobacteria that were captured by a host and reduced to metabolic slavery. So all photosynthesis, it's true, all photosynthesis that uses water as a source of electrons and generates oxygen is either directly or indirectly derived from cyanobacteria. So they are my candidate for the most important organisms in the history of the Earth. I once got into an argument with someone who liked turtles, but I think I have the better case to be made. So how much oxygen accumulated? Did we go from a world that was very different from ours to a world very much like ours? Well, a lot of people have worked on that. We worked on it. This particular diagram was published about 2 weeks ago by Tim Lyons in this department. And it says something that I think most of us would agree with, and that is, while on the left-hand side, before 2.4 billion years ago, there was essentially no oxygen. And for the last 500 million years or so, there was oxygen at least broadly comparable to what we have today, at 21% by volume. But that for most of the intervening 2 billion years, there was a little oxygen, but not much. Maybe a percent or a couple percent of the oxygen we have today. And under those conditions, the atmosphere has oxygen, surface waters have oxygen, but as you go into deeper water masses, commonly they will still be without oxygen. And again, I won't go through the geochemical lines of evidence that Tim, our group, and many others have looked at, but again, it has to do with how elements, isotopes, and minerals respond to the presence or absence of oxygen. So this great oxygen revolution 2.4 billion years ago produces an intermediate world, not like the early one, not like the later one. But it's a world in which we find now lots of fossils. What you see in that panel on the left is a series of cyanobacteria. On the right is, again, a so-called phylogeny, a hypothesis of evolutionary relationships among cyanobacteria based on molecular sequence comparisons. And what I tried to show there with those asterisks is that all of the groups of cyanobacteria represented by those red asterisks are already found in the fossil record by about 1,800 million years ago. So the kind of diversity of cyanobacteria we see today, and I should point out that we might think cyanobacteria are kind of nothings now because of plants and algae, but still more than a quarter of all the photosynthesis on this planet is done by cyanobacteria. And again, during the first part of the so-called Proterozoic eon, they really radiated and produced the kind of diversity we see today, and they're fossilized to prove it. At the same time, beginning around 16 to 1800 million years ago, about halfway through the known history of life, we start seeing fossils of things that belong to our own branch of the tree, the eukaryotes. Large, ornamented cells in modest diversity. And through time then, as we go upward through the Proterozoic in Earth's middle age, we find more and more evidence of eukaryotic diversity, more and more things that we can actually recognize. So that kind of nifty looking structure on the left, it's about 1100 million years old, comes from Arctic Canada, and that is unambiguously the fossil of a red alga. So we have now not only eukaryotes, we have photosynthetic eukaryotes, we have simple multicellular eukaryotes, and we have things that we can actually recognize as belonging to an important tree of life. As we go upward through time from there, we see that that kind of crazy looking thing on the bottom is one of an increasing diversity of protozoa of various sizes and shapes that become more abundant, suggesting that eukaryotic diversity, particularly after about 800 million years ago, is building up. In the upper right, where you see those two vase-shaped things, the left one is about 750 million years old. It comes from deep within the Grand Canyon. The structure on the right is the sort of shell, if you will, made by modern amoebae that make their own tests. And again, we think we can make some very close comparisons between living testate amoebae and fossils that appear in abundance and diversity about 800 million years ago. And then the things in the center are kind of interesting. One of my students, Phoebe Cohen, and I worked on these together recently. Phoebe has found thousands and thousands of small scales. Each of those scales would be about 30 microns in diameter. We know that a lot of single-celled eukaryotes make scales that armor themselves today. And Phoebe has found, something like 40 different kinds of these scales in rocks that are 800 million years old. So by 800 million years ago, I should say the one on the right is actually also a red alga from about 580 million year old rocks in China. So as we come closer to the familiar fossil record of animals, not surprisingly, we see a world in which eukaryotic organisms in general, the branch of the tree from which animals will emerge, are becoming more diverse, more ecologically important. And in fact, among the various groups that we think started to diverge around 800 million years ago are the animals. And at least based on molecular arguments and estimates made from comparative biology, it's thought that animals diverged from their protozoan ancestors about 750 to 800 million years ago. But as I already told you, we don't see much of a fossil record of them until we start getting large body fossils around 580 million years ago. So one of the questions you might ask is, why, what kind of world could support this kind of cryptic biology of animals, and then how would that world change in a way that would support big, fancy animals of the type we understand? And one answer to that is oxygen. If oxygen remained low 800 million years ago, but started to increase closer to 600 million years ago, that would allow for animals that were larger, that could be fossilized, that could be, have different functions. And again, this is, I won't take the time to explain this other than to say, this is again from Tim Lyons' group, and when you look at the amount of uranium in sediment, something you and I would not normally do, but Tim does, and what you see is that the last 580 million years ago look very different from the 3 billion years before that, and the explanation is that we are now moving to a world which is more similar to ours in terms of oxygen. For the first time, we are coming close to a world that you and I would regard as having breathable air. And there's an interesting linkage to that. What's we're now on the brink of what's commonly called the Cambrian Explosion, that is the geological manifestation of diverse familiar animal life that begins to unfold about 540 million years ago. And for years people have suggested that maybe that was the result of ecology, that as soon as you invent carnivorous animals, animals that eat other animals, that would actually start a kind of evolutionary arms race that would lead to diversification of both predator and prey organisms. Other people have said, well, no, what allows animals to really go to town is having enough oxygen to support different physiologies. And this argument, based on a paper that we did, but really the engine was Eric Sperling, a postdoc in my lab, suggests that those are actually two sides of the same coin. It turns out if you go and look in places where there's very low oxygen today in the oceans, there's plenty of animals. They're all tiny. They're not going to fossilize. One group of animals that is not present when oxygen is very low, a couple % of today's levels, is carnivores. So as oxygen began to creep up toward more modern values, we think that that actually made a whole new type of animal feeding possible, and that's carnivory, and that then would help facilitate what we see in the Cambrian period. And all of the things that you're seeing there are Cambrian animals, and you can look at them and you say, yeah, I see that. There's a mollusk there. There's an arthropod. If you're a little bit more specialized, you'll say, oh, there's annelid worms, etc. And that's really what you see. In the Cambrian, for the first time, we see these diverse animal body plans that will now characterize the oceans for the next 500 million years. So let's get to the end then. What I'm suggesting then is that through time and hard work on the part of a lot of people all over the world, we have been able to, to a first approximation, to fill in that deep history of life. Life began more than 3.5 billion years ago on this planet. It was at first life that lived in the absence of oxygen, mostly bacteria and this other prokaryotic group, the archaea. Only about 2.4 billion years ago do we start to have oxygen accumulating in ways that will make new types of life possible. That would result in both bacteria and archaea that use oxygen for things like respiration, and it would make possible our own group of life, the eukaryotes. Then there's a long interval of time when we have fairly static ecosystems, but again, the world changes in terms of oxygen availability toward the end of the Precambrian, gets slowly at least to oxygen levels like today, and then we add other new forms of life, the kind of animals that we're familiar with. So the idea then is that we can chronicle a deep history of life. Using the tools of chemistry, we can chronicle a deep history of environments, and as near as we can tell, those two things seem to change in concert. That is, there seems to be a coevolution between life and environments over the entirety of our planetary history. And so the penultimate slide then just gives the punchline that life is not this very active entity that is playing out on a static planet, that we live on a dynamic planet. It changes on various time scales. The positions of continents change through time because of continental drift. Climate changes, so we have ice ages episodically, and then they go. We know from measurements and models that the amount of CO2 in the atmosphere has changed dramatically through time. And then every once in a while, on the lower right, the solar system really plays a trick on us, and we get these large-scale impacts. So what you see there is two dinosaurs on what I can assure you is the last date they will ever have, looking out, watching this great meteorite hit the Yucatan Peninsula, which caused one of the great mass extinctions. So what I'd like to argue then is that it's actually inappropriate, and this is maybe an advertisement for your new institute, which I love this idea very much. It's actually inappropriate to talk about the Earth without thinking about life, and it's inappropriate to talk about life without thinking about the Earth, because they're so intricately intertwined today and always have been. And that leads me to the final slide, which is that we live today in a time of potentially geologically important rates of environmental change. And if you look, this is a very interesting slide that comes from Jim Barry at Monterey. And what it doesn't show the amount of anything, but it shows over the last 400,000 years the rate of change of CO2 in the atmosphere. And you can see not much is happening for nearly all of that until you get to the industrial age, and it shoots up. The one on the right shows rates of change in the pH of the ocean, something that you've maybe heard about in ocean acidification. Again, there's a little bit of wiggling back and forth, but boy, oh boy, do things change in the 20th century and will continue to change in the 21st century. So what I'd like to leave you with is that just as life has responded to, both caused and responded to environmental change in the past, one of the great challenges, I think, for our children and grandchildren is that they are going to live at a time when the environment is changing, and that will have biological consequences. In this case, maybe there's cause for optimism because there is a biological agent of that change. It's us, and we can choose to try and make the world the best place we can for our children if we choose to do so. So thank you very much. Questions?
So we will have time for a few questions, and how we're going to do this is if you raise your hand, we have a couple of science ambassadors and others who will nip to you with the microphone. So you're asking a question in the microphone. So
I don't need a microphone.
Oh yeah, but the other people over there can't hear you. So we're going to. I've been told we're definitely going to keep with the microphones. So if you can.
Testing.
Okay.
How were the microbial reefs built?
Good question. The question is, how are these microbial reefs built? And this is something we can actually observe today, that you make a microbial mat, and then perhaps a storm will bring in just some fine-grained carbonate sediments, carbonate mud or silt. And what happens is the microorganisms will actually grow up through that silty coating and then cover its surface and make another layer. So they are actually trapping and binding sediment, and then actually microbial processes deeper in the mat will cause carbonate minerals to precipitate and cement that sediment. So this is something where the mat organisms are actively interacting with the sediments, causing this buildup, and that's what we actually see in that laminated structure. It is a record of a series of surfaces that build up through time because of the formation of the mats and then trapping sediments. Does that make sense?
Yeah.
Good, then I'll stick with it. Okay. Yeah, just tell me.
Must I wait?
Yes.
Okay. Is this entire story marine, or are there some bits and pieces in the much less abundant continent?
That's a very good question. I have told this as a marine story, and the question was, is there a non-marine chapter to this story? It turns out that the answer is most probably, but we actually have relatively few sedimentary rocks that we can identify as non-marine in the deep past. We do have lakes that go back at least to 2.7 billion years old, and they certainly have microbial ecosystems in them. They have stromatolites, they have the kinds of chemical signatures of life that we see. But, you know, one of the things you have it's kind of interesting to think about in terms of life on land versus life in the sea. Most organisms that live on land live in places where there's net erosion of sediments, and many organisms that live in the shallow sea live in a place where sediments are on average accumulating. And for that reason, we tend, even in the last 400 million years when we have plants and animals, and there's an extraordinarily important component on land, is that the land record tends to be much less complete than the marine record. And certainly before we have land plants, it is much, much harder to interpret. So the bottom line is that, yes, we think that microbial life covered the land surface very early, just as microbes can make mats on the land surface today, but the actual documentation of that is very fragmentary.
He'll bring you.
Perhaps you explain this, I'm not clear. You talked about cyanobacteria
Yeah.
providing the oxygen.
Yeah.
But were you saying that they were mostly in the CO2 atmosphere, whatever atmosphere, and,
Yeah.
and how did they begin, if they were in that atmosphere, to produce it, the oxygen?
Oh, okay. So the question is, what are all these cyanobacteria about? What are they actually doing? And how could they take hold in a world without oxygen and generate a new world? Is that a fair statement of the question? Well, it's interesting. It turns out that if you look closely at the bacteria, there are at least 6 different groups of bacteria that are capable of photosynthesis, of which cyanobacteria are only one. The rest of them, to do photosynthesis, you actually need to find a molecule that you can break down, because you need electrons to take carbon dioxide and make it into a sugar. And so all the other bacteria get their electrons from things like hydrogen sulfide, that rotten egg smell gas. Some get it from hydrogen gas. Actually, iron in its soluble form can provide electrons. And we think that on the early Earth, the first photosynthesizing organisms were actually using these kind of compounds. It turns out one of the hardest biochemical reactions we know is the enzymatic breakdown of water to provide electrons from that. The advantage of using water is it's everywhere. The disadvantage is that it takes a lot of energy to extract electrons from it. The consequence of using water rather than sulfide or hydrogen is that the byproduct is oxygen. So we know, and you can do this in the chemistry lab, that the oxygen produced by photosynthetic organisms like plants comes from water. And at that point you have 2 issues. One is, how do you keep from being harmed by oxygen? And all organisms have molecules that can actually quench reactions that might damage them from oxygen. And then is, how can you learn to use the oxygen once it's there? And there's, again, a big literature on the evolution of enzymes that shows how once oxygen is present, then it can be used actually to break down organic matter. That's what all of us are doing now. So I'm not sure whether I've answered your question, and please tell me if I haven't. But we know from comparative biology that there are various steps in the evolution of the complex photosynthesis we see in plants. Much of those steps are still represented by organisms today, and in a sense, organisms that use water ended up making life difficult for the other ones, because as soon as you start generating oxygen, that will actually get rid of all the other compounds you could use for your electrons. So it's oxygen producing photoautotrophs. They're kind of like kids in the sandbox that knock everybody else out and make life safe for them. Does that get at your question? Okay. Any other questions?
Okay, well, that's great. Well, all of you can join me in thanking Ambient.